Hello everyone and welcome to the vlog. I am Kirkis R. Montealto from STEM 1205 St. Patrick. I am 17 years old and a grade 12 STEM student from Notre Dame of the Liangas University. For today's video, we are going to do a 3D diorama of an animal cell as a part of our performance task in General Biology 1. So without further ado, let's get started. Here are the materials that we are going to use for this performance task. Cardboard, paper saucer, scissor, markers, yarns, cartolina, glue guns, and glues that unfortunately not shown in this clip. So, here is me cutting the cardboard into circles to form a semisphere for the base of my organelles. I just cut the edges and formed it into a circle because later on the circles will be used to form a semisphere for our organelles. So basically, I just cut 4 circles to form a semisphere. And here are the 4 circles that I will use to make a semisphere. After I cut the circles, I paste its stand for it to be able to stand on its own. I used double-sided tape for this. I covered the semisphere light yellow that serves as our cell. And I used darker color yellow for our cytoplasm. Ta-da! So here is our cell without its organelles. After making the cell where we will going to put our organelles, I made use of cartolina as our material to make an organelles, such as the centriole, smooth endoplasmic reticulum, rough endoplasmic reticulum, mitochondrion, lysosome, Golgi apparatus, and more. So my niece begged me that she will help me to do my project for this General Biology 1. So in this part, I am using orange marker to color the ribosomes. So now I am using glue gun to stick the part of the organelles and paste it in the cell. I used yellow yarn to be our plasma membrane. After pasting the plasma membrane, I now arrange the organelles on where I should put them on the cell. Ta-da! We are now done in making the animal cell. So unfortunately, we are having a power interruption and I cannot continue it because it's already late. It's 10:38 p.m. I'm going to update you tomorrow for the explanation of the parts and the functions of the cells. The nucleus is the control center of the cell. It contains the cell's hereditary information, or the DNA, controls the cell growth and reproduction. Inside the nucleus, there is a nucleolus that helps to synthesize ribosomes by transcribing and assembling ribosomal RNA subunits. The rough endoplasmic reticulum has a ribosomes, which are small, round organelles whose function is to make those proteins that are very essential in a cell. The smooth endoplasmic reticulum functions in many metabolic processes. It synthesizes lipids, phospholipids as in plasma membranes and steroids. Centrioles play a role in organizing microtubules that serve as the cell's skeletal system. The plasma membrane regulates the transport of materials entering and exiting the cells. Vacuole is a membrane-bound cell organelle. In animal cells, vacuoles are generally small and help sequester waste products. Mitochondria, the powerhouse of the cell, generate most of the chemical energy needed to power the cell's biochemical reactions. Cytoplasm is the gel-like fluid inside the cell. All of the function for cell expansion, growth, and replication are carried out in the cytoplasm of a cell. Golgi apparatus or Golgi complex function as a factory in which proteins received from the ER are further processed and sorted for transport to their eventual destinations. Lysosomes are membrane-bound organelles with roles in processes involved in degrading and recycling cellular waste, cellular signaling, and energy metabolism. Ribosomes have two main functions: decoding the message and the formation of peptide bonds. They can be found also in the rough ER. So that is it for the video, guys. I hope you enjoyed and learned something from this video. I am Chris R. Montealto from STEM 1205 saying keep safe, keep sane, and stay hydrated.